Okay, so welcome everyone to this webinar hosted by Biochemical Society and Portland Press, which is part of our Biochemistry Focus webinar series. So topics in the series include different research areas in the biosciences, as well as practical sessions to support career development as well. So you've got an opportunity here to ask questions via text, or we welcome suggestions for future topics as well, or if you yourself want to do a talk in the webinar series, then please get in touch. So I'm Dr Connor Rogerson. I'm a senior postdoc at the University of Cambridge, and I'm the chair for today's session. So today we'll be hearing from Dr Tineke Lenstra, who will explore her lab's research into understanding the mechanisms that regulate transcription dynamics in single cells. So Tineke received her bachelor's and master's degree in biomedical sciences from Utrecht University. She then completed her PhD in 2012 in the laboratory of Frank Holstiger at the University Medical Center Utrecht, where she used genome-wide expression analysis to study transcription. She then moved to the laboratory of Dan Larsen at the NCI to complete the postdoc, and then established an independent group in the Netherlands in 2016. Her lab focuses on the regulatory mechanisms of transcription in eukaryotic cells, and for her work she's received a number of prestigious awards, including Fellows Award for Research Excellence and the NCI Directors Innovation Award. So Tineke, over to you.
Okay, thank you, Connor, for the nice introduction, and thank you for inviting me to give this webinar. I'm very excited. So our lab is really interested in understanding transcription regulation using single cell and single molecule approaches. And for this webinar, I'd like to give a brief introduction into the field of transcription dynamics, and then also show some of our latest work. So why do we care about transcription regulation? Well, gene regulation is essential for multicellular life, where every cell in your body has the same DNA, yet different cells have different shapes and functions because they express different proteins, and these are the molecular machines of your cell. And so transcription regulation ensures that the right gene is activated in the right cell type, which is then transcribed into RNA to eventually get translated into proteins. And so the ability to form these different cells and to activate the right gene in the right cell requires proper regulation of gene expression. And over the last decades, there has been many studies on what proteins are involved and how they interact. And so we have this basic understanding about some of the components that are required to activate genes. And I'll show you, I'll walk you through the schematic of what we think so far. So we know that in front of genes, there's regulatory regions called promoters, but also more distally in the genome, there's regulatory regions called enhancers. And these contain motif sequences that can be recognized by specific proteins called gene-specific transcription factors. And these can then recruit other proteins called cofactors, such as mediator, but also chromatin remodelers that can slide or evict nucleosomes that normally wrap the DNA, and that can make the DNA more accessible for other proteins to bind. And one of these protein complexes that can then bind is the pre-initiation complex at the core promoter. And this then allows the assembly of RNA polymerase at the gene, start initiating and then elongating the gene, producing RNA. And even though we have some basic understanding of these mechanisms, there's still many open questions. And in particular, we have very little idea about the dynamics of this process. And so we try to understand the single cell dynamics in order to understand the mechanism. And just to make an analogy why it's important to understand this dynamics. Let's say you're an alien and you're observing Earth and you really want to understand how a traffic light works. It wouldn't be very useful to get an average traffic light brownish color. That wouldn't tell you very much. If you would start looking at individual traffic lights, you would understand that there's 3 colors, but still you wouldn't really know how it works. In order to really understand this, you need to see the order of events, how traffic lights change their color and how the traffic responds to this. And the same applies to the cell. If we want to understand gene regulation, we need to understand how all these components dynamically interact with each other, how they change over time, and how this results in the production of RNA. And this is what we try to do. So just to give you a flavor of how dynamic this process is, here I'm showing you two example movies of two different of these processes. So on the left, I'm showing you transcription factors that are diffusing through the nucleus. And these dots that you're seeing are individual transcription factor molecules. And sometimes these transcription factors, they bind to DNA, and this is when they become immobile and when you start seeing this spot appear. But you also see this spot only lasts for a few seconds. So actually, transcription factors only interact with the DNA for a few seconds. On the right, I'm showing you this transcription of a single gene. Here we labeled the nascent RNA of a single gene. So every time this gene is transcribed, you see this spot appear in the nucleus. But you also see that this spot changes in intensity and actually sometimes also disappears. So also the production of RNA itself actually is highly dynamic. So even though this cell type should be expressing this RNA, it's not like it's continuously producing the same amount of RNA each time. And that means that transcription by itself is inherently a stochastic process. And what do I mean with this? What I mean with this is that RNA polymerase, it needs to initiate on this gene, but the exact moment that this RNA polymerase initiates is determined by the random motion of molecules. It interacts with things and then it goes to the promoter, but we can't exactly predict at what moment this occurs. And so if we plot the number of initiation events over time, here indicated by these green lines, we can see that there's slight variation between these events simply because of the molecular motion of all the molecules in the cell. If we then plot the number of RNA in the cell, then sometimes we have a little bit of more production when these events are closer together, and sometimes we have degradation of an RNA. So there is a mean expression in the cell, but there also are fluctuations around the mean that come from slight variations in the production and degradation of this RNA over time. So if we then start to look at multiple cells and we thought they all have these fluctuations and you would look at a single time point, that means that maybe in the upper cell, in this moment in time, you slightly have higher levels of RNA because you just produce some RNAs. The middle cell maybe just happens to have a little bit of lower RNA because it degraded some, and the third cell maybe has average or intermediate levels of RNA. And so in a population, you will find all these examples that all have slight variations coming from the fluctuations in transcription. And if you could then count how many of all of these cells you have that have low or high, you can make a frequency distribution of the number of RNA or even the number of protein per cell that simply come from the fluctuations in gene expression. And when we're measuring RNA expression in a tissue or in a cell line with qPCR or with RNA-seq, you usually measure only this average. But in real life, all these cells are actually slightly varying, and so they will have a certain distribution coming from some of this coming from the fluctuations. But this is not the whole story. There is the stochastic transcription, but on top of this fluctuations coming from the initiation of polymerase, we also found, or actually many groups found, that transcription often occurs in bursts, where instead of initiating single polymerases, actually polymerases often occur in groups, and then there's a period where there's no initiation, where the gene seems to turn off, and then again several polymerases initiate. And so there's more of a situation where this gene switches between different states, some which are transcriptionally permissive, there's loading of polymerases, and then the gene can actually turn off. And this bursting property has been observed for many different organisms, so it seems to be a conserved property. It's been found in bacteria to budding yeast all the way to mouse and human. It doesn't occur for every gene. There are also genes that don't show bursting, especially in, for example, budding yeast. But for mouse and human, almost any gene that has been looked at shows this bursting behavior. So it seems to be at least some inherent property of transcription itself. Yet the origin and the regulation of bursting is still largely unknown, and this is what our lab is really interested in. What we know, though, is that if genes show this bursting transcription, then their transcriptional fluctuations become much larger, because now every time they produce an RNA, they produce several, and also there's longer periods where they're off, so they also go down more. So the fluctuations in transcription are larger than if you would have constitutive transcription, and this also then increases the heterogeneity in the population. And this heterogeneity actually depends on the parameters of the bursting, so on how many polymerases and the time between bursts. So if you, for example, have a cell that shows bursting transcription for a certain gene, and it would increase transcription twofold, then you can either increase the size of the burst, so the number of polymerases that initiate during a burst, or the frequency of these bursts. And even though these result in the same mean change in expression in the population, they have very different effects on the heterogeneity, where increasing the burst size results in much more cell-to-cell variation than increasing the burst frequency. Yeah, we don't even really understand if cells use this mechanism of regulating burst size and burst frequency to actually have a way to tune the heterogeneity in the population. I think that's still unclear, but perhaps it's just a consequence of the fact that genes need to be regulated, and this creates this bursting transcription and regulates the size and the frequency, but it could also potentially be a way to tune the heterogeneity. So I told you that this bursting can create variability. Here I'm showing you an example image of single molecule fish where we hybridize probes to individual RNAs of a certain gene. And then you can also see this is in a genetically identical cell line, that some cells have virtually no RNA and some cells have very many RNAs, as indicated by all these dots, and also the bright transcription site where many RNAs are being produced. So some of this heterogeneity we see comes from the stochastic transcription and the transcriptional bursting I just talked about, but obviously there's also other forms of sources that can contribute to this heterogeneity, some of which are, for example, the size of the cell, where larger cells usually have more RNA. Different cell cycle stages can have different levels of transcription, but also slight variations in the level of RNA polymerase or ribosome or other regulatory components can affect the heterogeneity in the population. And so this all contributes to this heterogeneity, but the transcriptional bursting and stochastic transcription also contributes to this. And you can distinguish between these different forms because the first one, the stochastic transcription, which is usually referred to as intrinsic noise, is inherent in the system, and so it's also specific to which of the alleles you're looking at. So if the cell has two alleles, then when polymerase will land on the gene differs for one allele versus the other. Whereas for heterogeneity coming from cell size or cell cycle stage, that is the same for both alleles inside that single cell, and so those two alleles will then show the same response to those changes. So why do we care about this heterogeneity? Well, it's been shown that the stochastic cell-to-cell variability can influence essential cell-fate decisions, and I don't think the full scope of what exactly it can regulate is known, but there are some really nice examples, and I'll just highlight two where it can affect, for example, here, differentiation. This is a really nice study from the Weinberger lab, where they showed that if you increase the transcriptional fluctuations genome-wide, then this makes cells more plastic to transition into new states, probably because it's easier to then pass a barrier. And so in ES cells, in 2D culture, when you increase the transcriptional fluctuations, these cells can actually differentiate faster than in regular conditions. Whether this is also something that occurs during embryogenesis, for example, that still remains unknown. It also has impact on disease settings, for example, in cancer drug resistance. There's some really nice work from Arjun Raj lab showing that there's these transient cell states that have this transient high expression of drug resistance genes, and that this can arise perhaps because of transcriptional fluctuations, and that at the moment you add drug, that these cells can now enter a different state and maintain in this high transcribing state to remain drug resistant. But the initial formation of these transient states is likely not through genetic sources, but through fluctuations or changes in the cell that are transient. So our lab is really interested in trying to understand the mechanisms behind these fluctuations, what regulates the transition between the on and off state, what is the molecular composition of these states. So in order to dive more into the mechanisms of how this is regulated. And in order to do this, we want to directly visualize transcription as it occurs in live cells. For this, we use a trick developed by Rob Singer's lab, referred to as PP7 or MS2. Here we introduce repeat sequences into an endogenous gene of interest. And when these are transcribed, they form these stem loop structures. And then we co-express in the cell a co-protein that is fused to a fluorophore and that specifically binds to these stem loop structures. And this results in the appearance of a dot in the nucleus, of which the intensity is proportional to the number of RNA that's being produced. So now we can directly follow the number of nascent transcripts over time inside living cells. We do this technique in our lab both in budding yeast as well as in mouse and human cell lines, but for today I will focus on our results in budding yeast. And we use this because this is a single cell eukaryotic organism that has many of the features that also are regulating transcription in human cells. But since we really don't understand much of the basics of transcription, yeast is a good organism because it's a little bit more basic in its regulation, and so it allows us to really identify some of the core components that regulate this transcription kinetics. So over the years, using these techniques, we tested several angles of what could regulate this transcription kinetics, and we started with the transcription factors, because these are the proteins that recognize the motif sequences and that tell the cell which genes to turn on. And as I already told you, these transcription factors, they bind to the DNA, but they are actually binding very transiently, so only a few seconds. So they also come off very quickly. And so at a single gene, a binding of a transcription factor may look like this, where it binds for a few seconds and then it also releases. And this transient binding has been observed for a while by many labs. And one of the major outstanding questions was, how does this transient binding robustly activate transcription? And one related question is, how does this few second binding actually result in these fluctuations in transcription? And what's interesting is that they are actually on very different time scales, where transcription factors only bind for a few seconds, but transcription can last for, these bursts can last for minutes to hours. So how can we disconnect these time scales? And in literature, there were several hypotheses about how these timescales could be connected. And in the first model, it was thought that maybe when we're imaging transcription, these transcription factors, maybe the way we're doing it, we're not measuring all the transcription factor. Maybe we're missing a subpopulation that has a very long residence time, and that these long-bound factors would actually turn on transcription. And when they release, this would end the burst and turn off transcription again. And in the second model, it was thought that maybe this transcription factors indeed bind very shortly, but then they are required to initiate a number of downstream steps that then would keep transcription going even after the transcription factor has already left. So the transcription factor would start the process, but you wouldn't need continuous occupancy of the transcription factor to keep transcription going. And in the third model, we thought that perhaps each of these transcription factors bind very shortly, but then they continuously exchange to have a longer period where there's high promoter occupancy. And then all these factors, when they would release, then this would result in an off period of transcription. And this model invokes some form of cooperative binding, where either you bind all together or you all release, so you get this bimodal occupancy of transcription factors, and that the chance of transcription factor binding is higher when there are already transcription factors bound to that locus. So far, these models were out there, and so far we were able to test or to measure transcription factor binding in single cells at single molecule resolution, and also transcription in single cells at single molecule resolution. Yet so far, it was technically very challenging to do both in the same cell. And so far, we couldn't really test any of these hypotheses. And so in our lab, we developed a novel technology that allows us to track a transcription site in 3D in a single cell, imaging a single plane, and then simultaneously measuring the transcription factor binding dynamics to a single gene, so not all the genes, but only to a single locus, and then seeing what the functional effect of that binding event is on transcription activation of that same gene. And for this, we chose the GAL system in budding yeast. We chose the transcription factor GAL4 because it's naturally very lowly abundant. And so that means that we can label a larger proportion of the transcription factor and have a high chance that it actually binds to our locus of interest. And what's also very nice about this transcription factors is that it has very few target genes. So again, that increases the chance that it actually goes to our gene of interest. We chose the GAL10 gene because it has 4 binding sites, and most other genes, or all other target genes, only have 1 or 2 binding sites. So again, then we hope to see binding of this transcription factor. And so we labeled the GAL10 target gene with the pp7 loops to follow its transcription, and the GAL4 with a halo tag that's not fluorescent by itself, but then we add this bright dye to the cells to label part of the molecules and see how they bind to the locus. So this is an example movie where we track the transcription site of GAL10 in 2D, and then we keep it in focus with an active feedback loop. And at the same time, we can see binding of these GAL4 transcription factor molecules to this locus. And now we can start to address what is the functional effect of these binding events on transcription activation. So then we get traces such as this, where we quantify the amount of binding over time in purple and the transcriptional activity of the GAL10 gene in green. And here, what I have to say is that we estimate that we label roughly 20% of all the GAL4 molecules. So that means that if we see that there is this binding of GAL4, that can mean that there's binding of 1 or multiple molecules. But if we don't see binding, that could be that there's no binding, but it could also mean that there's binding of unlabeled molecules. So to analyze this data, we first binarized it to know when there's binding and when there's transcription activation. And then we wanted to know, do we even see specific binding of the transcription factor to the single target gene? So we plotted the duration of the GAL4 binding events, so the residence times. This is the survival fraction. And what you should get from this curve is that there seems to be these 2 slopes, meaning that there's 2 populations of GAL4 binding to this gene, one that has an average binding time of around 20 seconds, and one that binds almost 1.5 minutes. And if we do the same experiment at a negative control gene that doesn't have any GAL4 binding site, we only see that there's binding for roughly 20 seconds. So that means that at GAL10 specifically, we find this binding population that's much longer than was previously observed, suggesting indeed that the way we were imaging before, we are missing certain binding events that are indeed much longer than we thought, but also that that we actually do pick up very specific binding to the GAL10 gene. So now that we know this, we started to set out to test these different models, and we started with the second model because what's very different from this model with the other ones is that you don't need continuous occupancy of GAL4 to keep transcription going, but just to initiate it. So to look into this, we took all the GAL4 binding events and we aligned it to the start of the GAL4 binding event. And then we plot the average intensity of the GAL4 and the GAL10 transcription signal. And as expected, the GAL4 signal goes up because this is what we aligned to. But then we see that this is very rapidly followed by an increase in the GAL10 transcription. And here it looks almost immediate. If we image with higher time resolution, we see that there's about 1 or 2 seconds delay between the binding of GAL4 and the initiation of transcription. So it seems to be very, very rapid. And so it's a very efficient process. So GAL4 is required to initiate transcription, but what happens if we now align to the end of GAL4 binding? So now we see that this also rapidly results in a decrease in transcription, suggesting that if GAL4 leaves, transcription actually turns off, and that you do need continuous occupancy of GAL4 to keep transcription going. So this would not be very consistent with the second model, where continuous presence would not be required. So next we tested the third model, where there seems the suggestion was that there would be this continuous exchange of transcription factors. So to analyze this, we took all the bursts and we overlaid them, and then we divided them into 5 time periods: before the burst, at the start of the burst, during the burst, at the end of the burst, and after the burst. And first we looked at the average occupancy of GAL4 during this burst. So we see that the occupancy is highest during the burst, again suggesting that you need continuous occupancy to keep transcription going. And it's much higher than before or after, which is when we see some background binding. And the start and end are this intermediate occupancy, because this is likely when GAL4 arrives and releases. So when we start to look actually, when does GAL4 arrive? When does it associate? Here I'm plotting the association frequency. And this is highest at the start of the burst. And this makes sense because if GAL4 arrives, then this usually starts a burst. But what's interesting is we also find very high association frequency during a burst. So even though transcription is ongoing, we see the arrival of new GAL4 molecules. What happens to the dissociation frequency? Well, that shows very similar things, where the dissociation frequency is highest at the end of the burst, again as expected, because if GAL4 leaves, this usually ends the burst. But we also see very high dissociations during the burst. So that means that during a burst, there's this continuous arrival and release of new GAL4 molecules. And this suggests that there is indeed this transcription factor exchange. So our data would be consistent with the third model where there would be exchange. But then we wondered, are there also molecules that maybe that they bind at the beginning, they stay bound throughout the entire burst, and then they release? But because we also saw this subpopulation with very long residence time. So now we re-image the cells with lower labeling density. So now we were really sure that we're seeing single GAL4 molecules. And then we calculate the percentage of overlap of these binding events with a burst. And then we do find that that there are bursts that completely overlap with a transcription factor binding event, but this is only about 16% of the burst. And for a long burst, this percentage drops to only 3%. So it looks like there are bursts where there's complete binding events overlapping, but this is not the majority of the cases. And likely, especially for the long burst, you need this continuous exchange to prolong the transcriptional burst duration. So to look at this a different way, we divided the burst into short burst and long burst, and we plotted the number of binding events of GAL4. And indeed, for short burst, we see much less binding events than for long burst, suggesting that for long burst, you do need this exchange. And you also see way more binding events than, for example, the for a negative control gene. So we do find this subpopulation that binds throughout the entire burst, but we think that the majority of bursts are coming from this transcription factor exchange. But so far, I've only shown you that there is this exchange, but this is really required to have these longer bursts. And so what we thought to do is to reduce this transcription factor exchange by decreasing the concentration of GAL4. So then the arrival of new GAL4 molecules should be decreased, and this should then reduce the burst duration. And indeed, this is what we see. If we can compare wild type to a situation where we have much less GAL4 concentration, a much lower GAL4 concentration, we see now that the duration of the burst also goes down quite a lot. So what I showed you is that genes with multiple binding sites, their transcription factors individually, they can bind quite short, but then they can continuously exchange to increase the period during which there's high promoter occupancy and during which there's a burst of transcription. And this high exchange and this also suggests that there's a higher probability of GAL4 to arrive when there are already molecules bound. That suggests that there's some form of cooperative binding where they all bind together and they all release together. And it's been previously shown for GAL4 using in vitro and in vivo approaches that it does seem to bind DNA cooperatively, although we really do not understand the mechanism of this cooperativity yet. But this cooperativity also seems to occur for many more transcription factors. And so it could be that this mechanism that we found of this transcription factor exchange could be a more global mechanism by which this short transient binding can result in robust transcription activation for many more transcription factors. What's also interesting is that if now the transcription factor concentration is reduced inside cells, that means that not only the frequency of the burst change, right, the probability of entering a burst depends on how often it binds, but also the duration of the burst changes. And so that means that in varying concentration, the burst size and burst frequency, they change together. And this has also been shown by the Greger lab during Drosophila development, that this seems to occur. So maybe this could be a mechanism that can enable that. But that could also mean that you can keep the noise constant in the if you change the transcription factor concentration, that the noise over the mean is now the same for that particular target gene. What we also think is that many regulatory regions in mammalian cells, like these enhancer regions that I showed in the introduction, they often have many binding sites with many different transcription factors. And it's not really clear how exactly they enable gene transcription, but perhaps this transcription factor exchange may also be a mechanism there to enable robust activation at enhancers. So I showed you that transcription factor binding dynamics are a very important player in regulating the fluctuations of transcription, but that their short dwell time requires continuous exchange to result in larger bursts of transcription. In addition to the dynamics of the transcription factors, we also started to look in the spatial organization. Because for many transcription factors and factors of the components of the transcriptional machinery, it was shown that they're not homogeneously distributed through the nucleus, but they occur in these areas of very high local concentrations that I'll call clusters. And there was an attractive hypothesis in the field that maybe these clusters of high local concentration, when they're near a target gene, maybe they could result in the loading of several polymerases, resulting in this burst of transcription. And when this cluster dissolves or moves away, then this would end the target gene transcription. And so we started to look at this for GAL4, and indeed we find that GAL4 also shows this clustering behavior. And we see that this clustering can help enable to move of GAL4 molecules to go to the target genes when molecules are already bound. So it seems to enable the target search process, where transcription factor maybe have an easier job finding the gene that they're supposed to be regulated. But we find that the molecules that are in the cluster but are not DNA bound, they don't really contribute to transcription activation. And in fact, they maybe even compete for interactions with the transcriptional machinery and maybe inhibit it. So there's a tight balance in how this clustering affects transcription on the one hand, helping transcription factors find the right locus, on the other hand, maybe inhibiting transcription activation. And thereby we don't think that this bursting or this clustering per se is the main driver of transcriptional bursting, at least not for these GAL genes that we looked at, but they do seem to play an important role, perhaps in regulating this cooperativity as I showed. I don't have time to show you all the data, but it's been published and I'm happy to talk to anyone that's interested. One other thing that could regulate transcription is the fact that DNA is wrapped in nucleosomes, and this can prevent also the binding of transcription factors or even of the transcriptional machinery. And so the idea was that perhaps there are configurations in the promoter where maybe nucleosomes are evicted or moved to a position where polymerases and the transcriptional machinery can quickly land and load multiple polymerases, and then when the nucleosomes move back, that this can turn the gene off. And so in a recent study, what we tried to do is look how nucleosomes in the CAL10 gene affect the transcriptional bursting properties. And we found actually that different nucleosomes affect the kinetics of transcription quite differently. And that likely also reflects their kinetics inside the cell. So some nucleosomes have to be remodeled only once for transcriptions to activate from inactive to active conditions. And other nucleosomes need to be continuously remodeled and also then regulate the frequency of each of the bursts of transcription to make it a highly efficient process. We also found that this remodeling of nucleosome is actually requires cooperation between specific enzymes, the chromatin remodelers, but also the transcription factors and the transcriptional machinery itself. Again, I don't have time to go into much more details. What I want to spend the remaining part of my talk on is on the role of DNA supercoiling. So what is DNA supercoiling? So when we think about DNA, we often think about this double helix that was presented by Watson and Crick in the 50s. Yet DNA rarely occurs in this relaxed form inside living cells, and that's because there's various processes that put tension on the fiber, such as DNA replication, DNA damage, but also transcription. And for example, for transcription, when RNA polymerase transcribes through the gene, it generates this overwound DNA, or what we call positive supercoiling ahead of polymerase, and underwound DNA, or negative supercoiling behind polymerase. You can kind of think of it as this old telephone cord that if it's all curled up and you put your fingers together and you move it apart and you create too little twist in the middle and too much on the outside. And the same happens to DNA inside our cells. And the supercoiling, it can actually feed back on the transcriptional process. So negative supercoil was shown to promote transcription initiation because it already helps unwind the duplex for polymerases to start transcribing. Whereas positive supercoils can help in the transcriptional elongation by removing DNA-bound proteins. However, too much supercoils can also inhibit both this initiation and the elongation of RNA polymerase. And so there's various enzymes in the cell called topoisomerases that quickly release these supercoils in order to prevent this transcription inhibition. Yet it was shown in a very elegant studies about 10 years ago that in bacteria, one of these topoisomerase enzymes is present in limiting amounts in the cell. And so at highly transcribed genes, there's so much supercoiling accumulation that this starts to inhibit both initiation as well as elongation. And this has to then wait for the arrival of topoisomerases and the release of supercoils before transcription can resume again. And so this accumulation and release of supercoils appears to be the main driver of transcriptional bursting in bacteria. Yet whether similar mechanisms of regulation also occur in eukaryotes was so far untested. In addition, we know from supercoiling mapping studies that supercoils can propagate along the chromatin fiber, at least up to 1.5 kb. And so that means that if a gene is transcribed and produces supercoils, that these supercoils can potentially travel to a neighboring gene and affect its transcription. So we wanted to understand how do DNA supercoils affect transcriptional bursting and how do they affect neighboring genes. And for this, again, we used the GAL genes in budding yeast. I previously showed you data on GAL10, but this gene is actually flanked by 2 neighboring GAL genes, also regulated by the GAL4 transcription factor called GAL1 and GAL7. And these genes are highly transcribed, so they likely generate a lot of supercoils, but they're also in different orientations. So GAL1 and GAL10 are in the divergent orientation, and that means that the negative supercoils likely accumulate in the promoter region and the positive in the gene body. And it was proposed that this negative supercoils in the promoter could result in enhanced transcription initiation of both genes, or perhaps a coupling of the genes and increasing their expression. For the tandem genes, however, the supercoils are thought to be negative, positive, negative, positive, and so there they alternate, and so maybe they even cancel each other out. So we wanted to understand how supercoils affect transcription of these genes. So first we wanted to see how does transcription of these neighboring genes even look like in wild-type cells. So we started with the divergent genes, and we used the orthogonal MS2 and PP7 system to label transcription of GAL1 and GAL10 in the same single cell. And so here I'm showing you an example movie where we can follow the fluctuations of both of these genes in the same single cell. And this results in traces such as this, where we can see that there's periods where both are inactive, periods where both are active, or where one is active and the other is inactive. So to see if transcription of this gene is somehow coupled or completely independent, we wanted to use an independent or an unbiased approach, and we chose the cross-correlation analysis. And this is not very intuitive, so I'll explain in a minute, very shortly what we do. So here are 2 hypothetical traces, and in a cross-correlation analysis, you calculate the correlation between these 2 traces, and then you shift one of the traces with respect to the other with different time delays, and you keep calculating the correlation. And you can shift the signal to the right, but then you can also actually shift it to the left, and then you get this negative correlation, so you can get a full cross-correlation function. And every time you see a peak or a valley in this correlation, that means that there's an enrichment of a depletion of the correlation compared to random chance. And the position of the peak and the valley gives you information about the time delay. So one always happens 10 seconds before the other, you will see a peak at 10 seconds. And the amplitude is information about the overlap. So this is hypothetical. If we now look at GAL1, GAL10 transcription, we start to see that there's a peak at 0 in the correlation. And that's the case to 0 correlation. It's roughly -100 and +100 seconds time delay. And 100 seconds is about the time that these genes require to do a burst of transcription. So what this shows is that GAL1 and GAL10 show coupled initiation, and then they both transcribe the gene, and then after that they become uncorrelated. We only observe this correlation when both of these genes are positioned on the same chromosome, because if we label 2 alleles on 2 different chromosomes, so the same 2 genes, now we see that they're completely independent and there's a flat line. So these 2 genes seem to show correlated initiation. We think a large majority of it comes actually from the transcription factor binding sites, where if GAL4 binds here, it activates both genes simultaneously. But if we start to now look at the tandem genes, GAL10 and GAL7, which don't share a promoter region, we still observe a weak peak at 0, and we don't really understand where this comes from. So this is much weaker than what we observe for GAL10 and GAL1, but still there seems to be some peaks suggesting that they do show some degree of correlated initiation. So in wild type, the GAL genes are temporarily coupled in their initiation. How does supercoils affect this coupling? So to address this, we wanted to perturb the supercoiling levels, and for this we use conditional depletion of the topoisomerases, the enzymes that release these supercoils. And in yeast, there's two that are involved in gene regulation, TOP1 and TOP2, that relieve both underwound and overwound DNA. And so we fuse them to an auxin-inducible degron tag that upon the addiction of auxin results in rapid degradation. And this system works very nicely, as in upon the addition of auxin, we get rapid degradation of TOP1 and TOP2. But we also observe that even if we don't add auxin and we just add all the components to the cell, we get some basal degradation. So we have now cells that have full levels of topoisomerases and intermediate and complete degradation of topoisomerases. So now what happens to transcription in these cells? Well, first of all, if we deplete topoisomerases, we see that many cells turn off. So apparently they are very important to regulate transcription. But we do also observe some cells where there's still transcription. And this is what the cross correlation looks like. And I'm just putting the wild type here as a reference. So we still observe this peak at 0, indicating that they still can initiate together. But now we see these really nice valleys at -100 and +100 second time delay. And I told you that it takes about 100 seconds to transcribe the gene. So what we think is happening is that when one of these genes transcribes, let's say GAL1, after it's done, apparently so much supercoils have initiated that by now it starts to inhibit subsequent initiation of the neighboring GAL10 gene. And also the other way around, when GAL10 transcribes, this starts to inhibit subsequent initiation of the GAL1 gene. We see the same for GAL10 and GAL7. So that means if the supercoils are not well dissolved, then all these genes start to inhibit each other in a time-dependent manner. What happens to the coupling? We addressed this using this measure called the transcriptional overlap, which we took from a publication from the Lushan lab, which is a measure for the transcriptional overlap that's independent of the transcriptional activity, because we also see lower activity in this strain. And what was surprising is that for both the divergent gene pair as well as the tandem gene pair, we see a drop in the transcriptional coupling of these genes. And this was surprising because I told you that people had suggested that if you increase supercoils, especially for the divergent genes, that this should enable maybe coupled initiation of both of these genes at the same time. Yet if we increase supercoils, we actually see a decrease in this coupling. And so we went into this in more detail, and I don't have time to explain you the entire mechanism or to go through all the data, but I can summarize the mechanism. What we found is that because of the architecture of this locus, where GAL1 and GAL10 are divergent, but there's this highly active GAL7 gene downstream only of GAL10, that GAL7 transcription seems to specifically inhibit GAL10 without affecting GAL1 as much. And this results in much lower expression of GAL1 and GAL10 compared to GAL1. And so this results in the decoupling of these genes. But then the question was, how do DNA supercoils actually inhibit transcription of these genes? And so for this, again, we went to single particle tracking of the transcription factors. So here we labeled single molecules, and we measured how long these GAL4 binds to the DNA. And this we didn't do in a gene-specific manner, so we just measured them at basically all the target genes simultaneously. And here we see that for wild type, we see much longer binding than if we start to deplete the topoisomerases, then this becomes much shorter. And we also see this if we quantify the average residence time of GAL4. So if the supercoils accumulate, then this seems to result in destabilization of the transcription factor, likely because the structure of the DNA is actually changed, and this changes the affinity of the GAL4 for its motif sequence. And maybe also other DNA-bound factors, such as the pre-initiation complex, become decoupled, and this is likely the driving force of reducing expression upon topoisomerase depletion. So to summarize what I showed you is that in wild type, we see that there's coupled initiation of the GAL genes, and that there needs to be rapid release of supercoils in order to maintain this coupling. Because if supercoils accumulate, if we deplete the topoisomerases, then we see that all these gal genes start to inhibit each other. And this likely comes from destabilized binding of the transcription factor. One thing that I didn't have time to show you about that we found is that many of the phenotypes that we found upon topoisomerase depletion, we didn't find in the wild type. And so it looks like the levels of topoisomerases in budding yeast are sufficient to prevent this transcription inhibition coming from the neighboring genes, but also from the genes on themselves, suggesting that at least in budding yeast in these conditions, that supercoiling is not the main driver of transcriptional bursting. However, supercoils are generated by many different processes, such as replication or DNA damage. And so it's interesting to think about how supercoils could regulate the dynamics and the levels of transcription of genes that are in the vicinity. So just to summarize what I showed you, we use single molecule imaging to visualize transcription and then the upstream process of transcription in single living cells in order to understand more of the mechanism. I showed you that transcription factors are a driving force in regulating some of the fluctuations in transcription, but that the dynamics of transcription are actually also regulated by the chromatin structure and, for example, by DNA supercoiling. And so to go back to the traffic light example, to understand the traffic light, you need to understand the dynamics, and the same applies to transcription. And over the last years, our lab, but also many labs in the field, have made a lot of progress in trying to understand some of these dynamics, but there's still a lot to be learned, and we're very excited in the future to continue these endeavors. So with that, I want to thank the people that did all this work. The transcription factor story that I showed you was mainly driven by a talented postdoc in the lab, Wim Pomp, with the help of Jos Meeuwse. The supercoiling work was driven by a talented PhD student, Hedda Patel, but she also had help from Wim, Stefano, and Ineke. But all of these stories were a true team effort, and was really excited to work with them. I want to also mention especially my collaborator, Domenico Libri, who showed for the supercoiling story that this mechanism of inhibition actually occurs genome-wide, so not only at the gal genes. I'm also very honored to work with very many collaborators in the field, and with the nice people at NKI that give a lot of feedback. If you're also excited by this, we do have some open positions. And I also just want to take 1 minute to highlight an exciting meeting that I'm co-organizing, an EMBL meeting in Heidelberg, where we're bringing together many experts that are also trying to use quantitative biology in order to understand the molecular mechanisms. So I really hope to see some of you there. I hope you enjoyed this webinar, and I'm very happy to take your questions.
Brilliant. So if anyone has any questions, please put them in the chat. And I think while people are doing that, I'll start off with my own question. So this is regarding the exchange of transcription factors. Did you ever look into what happens when you have two different transcription factors that regulate the same gene, and how the cooperativity between those TFs, sort of how one transcription factor could recruit the other and vice versa?
Yeah, that would be really cool if we could do that. I think that would be the eventual goal, to start mapping all these individual ones and then mapping how they interact, not only with other transcription factors, but also components of the transcriptional machinery. And not only actually also in yeast, but also in other organisms. So we're exploring some of these things. One thing that's challenging is that, right, these transcription factors have many target genes, and having a chance to see them at the right target gene, if you're only labeling a small percentage, you have to image many cells before you see the correct binding event. And we had this nice gal system where this seems to work. If we now start doing two different transcription factors, the chance of them seeing together and seeing enough events becomes even much shorter. So I think we there have to do some clever tricks to increase the number of events that we can observe, and we have some ideas how to do this, but we haven't done it so far.
Amazing, thank you. So in the chat, we've got a question about how does understanding transcription affect drug design, considering the implication of feedback enzyme activity inhibitors? How does this topic aid the management of a genetic disease? So probably how does this research on core transcriptional mechanisms sort of relate to disease?
Well, transcription is important for almost any disease, right? It's important for the maintenance of cells. But just to give an example, in cancer, there's often transcription factors are either they have fusions with other factors, they are overexpressed, and this changes the transcription of those cells, right? Enabling them also to proliferate faster, letting them become cancerous. Cancer cells also have to change their transcriptional programs to have metastasis or to even become drug resistant. And so all these things are then also important in those settings. We haven't really at the moment gone into how exactly, for example, the binding dynamics of transcription factors change if you add drugs, but I know there's groups that are working on this and using some of these assays in a high throughput manner to really beautifully read out what happens at the molecular level when you apply, for example, drugs to cells.
Yeah.
Does that answer the question? It was a pretty big question.
Yeah, I think so. Follow up for myself, because in cancer cells you often get extra chromosomal DNA amplifications, so like on basically intracellular plasmids. So I wonder if there's like deleting topoisomerase from cancer cells would actually be lethal, because I assume they would need topoisomerase to deal with those supercoils.
Yeah, I mean there is a topoisomerase inhibitor. It's a treatment for cancer, but I think it also it's difficult to say whether that then has effects on the transcription only, because topoisomerases are also essential for replication and to make sure that the strands don't intertwine when the genes are separated, the chromosomes are separated. So they are being used as drug for cancer. And it would be quite nice to actually know if, for example, those circles now have different transcription activity, but I don't think anyone has looked at that so far.
And then last question, is there a particular length of DNA where the generated supercoils of one's genes no longer affects the neighboring genes? So if you sort of put the genes further apart from each other, does the effect stop at a certain distance?
Yeah, we tried to test this. So we moved one of these genes more downstream to see when it would stop inhibiting the neighbor. And we could go up to where there would be an intergenic distance of more than 2 kb, and we still saw inhibition. So in literature, it was suggested that 1.5 kb was the limit by which it could travel. But even if we moved it more than that, we still saw inhibition. So we don't know how far it can go, because in yeast, the genome is also really tight, right? So most genes are really close to each other within a kb, so it's really difficult to really address. We did see genome-wide that the further you went, the less inhibition there is. But also in mammalian cells, the distances between genes and also between genes and regulatory elements is much larger, but we don't know actually how far they can travel. So maybe there is more communication than we think from the supercoiling. I think that's still an outstanding question.
Super. Brilliant. So I think we'll end it there in the interest of time. But Tineke, thank you so much for giving up your time to present us your data. It was fantastic. Thank you.
Yeah, thank you. It was very fun.